Well, we're back here, and now what we're going to do is look into the female reproductive system, which is the other part of chapter 26. When we talk about this, we have to deal with the various structures, both the external genitalia and the internal genitalia. We also have to keep in mind with the female that there is a mammary gland, which is usually considered part of the, although it doesn't involve immediate reproductive capabilities, it plays a role in a supportive sense, since the mammary gland produces milk to nourish the infant. Now, when we start looking at the internal genitalia, first off, you got to deal with the ovaries, which produce the oocytes. They also produce hormones, estrogen and progesterone. And then we have to deal with the female reproductive tract, also known as the female duct system. We talked last week about the male duct system, and now we're going to deal with the female. The female internal genitalia include the ovaries, the uterine tube, the uterus, and the vagina. Now, the female external genitalia include the vulva, which is kind of a collective term. It will consist of the mons pubis, the clitoris, and the labia, both the major and the minor. And you can see this here. Here we have the mammary glands up here, which are sort of modified sweat glands, interestingly enough. The external genitalia is down here, and then the internal structures here. The major ones are, of course, the ovary, the uterine tubes, otherwise known as fallopian tubes, the uterus, and the vagina. Now, major female reproductive organs are the ovaries, uterus, and their associated structures, which I've talked about. We're going to get more and more in depth with both structure and, to a degree, the function of them. When we talk about the ovaries, their functions are to produce immature gametes, oocytes, to secrete female sex hormones, estrogen. And when we say estrogen as a plural, it's not because it's a typo. Actually, estrogens are a collection of very similar molecules. They may have, for example, in one site a methyl group. If you can picture the cholesterol molecule, that four-ring structure, similarly, progesterone has that similar structure. They start off kind of like a sterol structure, but then you have extra extensions on some of the edges of the four-ring structure. So estrogens include estradiol, estetriol, estrone, and then of course we've got also progesterone. Finally, the ovaries produce inhibin, which is interesting because we dealt with inhibin in the male reproductive system. And really what this does is it provides a negative feedback control of the pituitary secretion of follicle stimulating hormone, FSH. We're also going to be looking into the uterus, the vagina, the uterine tube structures. When we talk about this, we have from the uterine tube, from the open end, we have the infundibulum, which on the edges have this fimbriae, which helps draw in a recently ovulated oocyte down into the tube structure, the ampulla, which is sort of the middle structure, and then the isthmus, which is right next to the uterus. Now, if you look in the anatomy from a, if you want to say from a side view, we're going to find these pockets that are formed alongside of the uterus. This is because we're down in the abdominal pelvic cavity. We have some of the structures there that have to be held without shifting greatly from one side to another. And so what I mean by that is you have these structures, the rectouterine pouch, which is formed between the posterior wall of the uterus and the anterior surface of the colon. And we have the Vesikko uterine pouch, which is formed between the uterus and the posterior wall of the urinary bladder. Because we have structures that are around there, we're going to get into also the ligaments that help stabilize everything. But I wanted you to be aware of some of these pockets and pouches, etc. We also have to deal with, of course, the external genitalia. The labia surrounds the central space called the vestibule. And then we have the clitoris, which contains erectile tissue that projects into the vestibule and contains sensory structures for sensations associated with the female orgasm. And as you can see here, here would be the Vesikko uterine pouch. And here on the other side, you have the recto-uterine pouch. Now, just to give you some perspective, here's the sigmoid colon right here. Excuse me, underneath the vesicouterine pouch, you have the urinary bladder. This may give you a little bit of an understanding when you hear some women who have talked about when they're getting into later months of pregnancy. Oh, that baby is just kicking on my bladder. Because if the child is moving around in this stretched out structure of the uterus, we'll get more into this when we get into development in the next chapter, they literally are sitting on top of what would be the urinary bladder. Okay, but you can see where you have the structures here of the thick uterus wall. You have the uterine tubes, which stretch out sideways here. The infundibulum sort of kind of caps superiorly the ovaries. Then you have, of course, the cervix at the edge or the end of the uterus, the vaginal fornix, you have the vagina here, excuse me, and you have also the external genitalia, primarily the clitoris, which is sort of vascularized erectile tissue, and then of course you have the labia, which we'll get more into in a little bit. As I mentioned a minute ago, we have ligaments. These are thickened peritoneal folds. They basically provide for support for these structures in the pelvic cavity. The first one is the suspensory ligament, which extends from the lateral surface of the ovary past the open end of the uterine tube to the pelvic wall. We have the ovarian ligament, which extends from the uterus near the attachment to the uterine tube to the medial surface of the ovary. The mesovarium is a thickened fold of mesentery, which supports and stabilizes the position of each ovary. And then we have the broad ligament. And the broad ligament is very much almost like a sheet. It's extensive mesentery that encloses the ovary, the uterine tubes, and the uterus. It attaches to the sides and the floor of the pelvic cavity, where it becomes continuous with the parietal peritoneum. Also, I want you to be aware of ovarian structure as shown in the next diagram, but just to give you a hinting, in this case, the ovary will have a hilum, which is sort of an entry point where blood vessels, nerves, they'll enter and leave from the ovary. This is the site where you have the ovary attached to the mesovarium. On the outer layers of the ovary, too, we get these two major divisions here, the cortex and the medulla. But when you get a little bit more fine detail here, we have the germinal epithelium, which covers the ovary surface and consists of columnar epithelial cells. We have the tunica albuginea, which is a dense connective tissue layer underneath the germinal epithelium. The cortex is the main area that you're going to have gamete production. So you're going to go through primordial and then primary, secondary, tertiary follicle stages. But the medulla, the inner core, is really composed more of blood vessels and nerves. And here we can see the ligaments here, the broad ligament, the suspensory ligament, as you can see over here. You can see also the ovarian ligament, which is really an attachment between the ovary and the uterus. And you basically will be able to see all of these structures here. Notice that you have also the mesovarium here, where you can see the uterine tube here. So you have these structures that prevent these reproductive organs from shifting around. They're not retroperitoneal. And so as the body moves around, they could shift around without the presence of these structures. You can see up in here the mesovarium here also. And then when we get into the actual structure of the ovary, you can see where you have the hilum coming in here, where you have more blood vessels that basically supply the nutrients, oxygen, removal of the waste, where you have the medulla, and then on the cortex is this area where you're going to have all these follicles going through various stages of development. You will have the outermost layer being a germinal epithelium, and then underneath that would be the tunica albuginea. Excuse me. Now let me talk about oogenesis. Now, if you have remembered your review of meiosis, great. If you have not, go back to that text on 1002 and 1003. Keep in mind that gametes, sperm and oocyte, are haploid. They have 1/2 of the amount of chromosomes for the organism. In this case, I used a very broad term. We're going to say human being. Fine. You and I have 23 pairs of chromosomes. In a sperm or an egg, it would have only 23 chromosomes, period. But you have the 23 meet up with the 23, the egg meets with the sperm, and now you have a fertilized egg, which we would call a zygote, and that has 46 chromosomes. Okay? Now, oogenesis occurs in the ovaries. Ovulation occurs during that 28-day ovarian cycle. We'll talk a little bit about that in a little bit. The term oogenesis, this is the formation and development of the oocyte. One of the interesting things that occurs between male and female when it comes to meiosis, the formation of gametes, is this. You start off with one cell that's diploid, 46 chromosomes. And by the time you're done going through the first meiotic division, the second meiotic division, you have 4 cells that are all haploid, have 1/2 of the chromosome. So in other words, they have 23 chromosomes each, 4 functional sperm cells. But not so with the female reproduction system. What you have is what is called an unequal distribution of the cytoplasm. So you get one egg that holds on to most of the cytoplasm. Now you might say, why? Well, we'll get into development later, but just to give you a short rendition, that egg, once it's fertilized, is going to contain a lot of the RNA and other necessary materials to start cell division. If you equally divided the cytoplasm into 4 separate oocytes, you would not get sufficient cytoplasm to sustain the developing zygote for the long run. Instead, what you have, because of the unequal distribution, is you're going to have one large oocyte, egg cell. The other cells that are produced get shortchanged, get cheated. They get small, tiny cells. They have very little cytoplasmic material, so they're called polar bodies. They're really non-functional. Even if a polar body got fertilized by a sperm cell, it would yield no viable embryo. They would die. Most polar bodies disintegrate later on. So when you have the first division, and you can see this in the next slide, you're going to have the first polar body that's formed. Now that polar body may or may not, depends on what text you read, go through the second division, but it's going to be one polar body making 2 more polar bodies, and they're both useless. But when we get into the second meiotic division, you're still going to have this large oocyte cell, and it's going to go through a second division. Some chromosomes are going to go into that polar body, but the other 23 chromosomes, boom, they're going to stay in that egg cell, and it's going to make it viable. Now, oogenesis has to start off with a female reproductive stem cells. Oogonia is the term used for a primary oocyte. Now, if you'll notice, I don't use the Roman numerals here to add to clarity here, but meiosis 1 basically is the process that starts off, and it proceeds until you get to the prophase of meiosis 1. Now, by the way, just to help you refresh, remember that mitosis goes prophase, metaphase, anaphase, telophase. Meiosis does the same thing, except it's reductive. So you go from diploid to haploid, 46 chromosomes to 23. So as a result, excuse me, As you go through the first set, they call that, prophase 1 or prophase 1 of meiosis 1, metaphase 1, anaphase 1, telophase 1, and then they don't stop. They go through a second division. Now, here's the only deal. It's easy to describe this in pen and paper. What happens in the actual biological system, such as the ovary, is a little bit more complex. Not that difficult. Just think of it this way. You start off with meiosis 1, and the process proceeds until prophase of meiosis 1. Halts right there. Everything's in suspended animation. When does it get going again? Puberty. You see, what happens at puberty is you're going to start having rising levels of that pituitary hormone we talked about early in the course, follicle stimulating hormone, FSH, which comes from the pituitary, as we said. It's under the control of GnRH, which is gonadotropin releasing hormone, which comes from the hypothalamus. Okay, keep that straight. When the rising levels of FSH occur in puberty, that triggers each month some of the oocytes to undergo further development, complete meiosis 1, and stop at metaphase meiosis 2. Okay, so at metaphase 2, at meiosis 2, in the metaphase stage, it stops. Now, ovulation can occur and the cell still is held in this state. Wait a minute. You mean the egg cell leaves and it's still stuck in metaphase meiosis 2? Right. Because what happens is it finishes up meiosis 2 at the time of fertilization. That's where the ovum undergoes meiosis completion, kicks out the second polar body. Goodbye. Meanwhile, the sperm cell is penetrating into it and it's bringing in its 23 chromosomes with the ovum's 23 chromosomes, and you've got a fertilized egg, a zygote. That's the term for a fertilized egg. Okay. Now, ovarian follicles are going to be these little cells around, sort of like nurturing and helping the development of the oocyte. And they're going to be these structures that are going to be present. They're going to be present at meiosis 1. Actually, if you take a look at some of the early micrographs there, you have what is called an egg nest. Not like we would see in a bird with a nest in a tree, but they kind of use that terminology. This is where you have primary oocytes. They're located in the outer portion of the ovarian cortex. They're inactive, though, but they're surrounded by a simple squamous layer of follicle cells. This is referred to as a primordial follicle. Now, primordial follicle will move into the next stage, primary ovarian follicle. That's when follicular cells start to divide. They become granulosa cells. And you can see, as we've gone through this meiosis 1, meiosis 2, you can see where you're stuck there. Meiosis 1 at the prophase state, meiosis 2. Remember, I talked about how you eventually complete the rest of meiosis 2 by the penetration of the sperm cell. Now, let's go back a second. I just started talking with you about the whole development of the follicle. Now, what's going to happen now are these follicles are going to go through the process. And as they go through the process, we're going to have these supportive cells that are going to provide nutrients and growth factors and trigger some of the genes inside of the ovary. The follicular cells are going to become the granulosa cells. And also what you're going to see, interestingly enough, is a special thin layer of cells, a region surrounding oocyte called the zona pellucida. That's actually going to be around to help basically prevent multiple sperm cells from penetrating the cell. Only one gets to be the fertilizer. Everybody else kind of wiggles around and tries to, but eventually we're going to talk about that later on, how you have a situation where you prevent having multiple sperm fertilizing that egg. Now, I've talked about the zona pellucida, but then we've got this other layer. What happens is some of the granulosa cells develops further, and they become what's called thecal endocrine cells. Now, these are the ones that are around the follicle. They're going to be producing, though, estrogens. So we move from now a primary ovarian follicle, and we're going into the secondary ovarian follicle. This is formed as the deeper layers of the follicular cells begin to secrete fluid. And this is going to occur and build up in small pockets, but later the pockets will all fuse. They're going to be filled with a fluid, and they're going to be called basically the antrum. The antrum is going to be loaded with follicular fluid, which contains hormones and a lot of other factors. And then we move into the tertiary ovarian follicle. By the time you've hit that, you're about day 10, 14 of the ovarian cycle. There is one thing. If we started with a bunch of different ovarian follicles, some move faster in development, others don't. Eventually one gets to, shall I say, the finish line, where it is going to become a mature tertiary follicle, and that follicle also goes by the name Graafian follicle. Here is where you're going to have some granulosa cells around the oocyte form this kind of protective layer. It's known as the corona radiata. And what it's going to do is it's going to stay around to a point even after ovulation. Okay, ovulation occurs. Bang. What literally happens is that what appears to be almost like a bump on the surface of the ovary will eventually rupture. And what happens is, for some women, they will actually say they felt that. It's called the Mennerschmeiß. And it's almost a painful situation for some, or some will say, they felt this little pinch. But for some women, they won't say they felt anything. But anyways, here's the deal. Tertiary follicle that's released is relatively a mature oocyte, except it's got to finish up that second meiotic division. It's going to have with it some cells that are going to support it, the corona radiata. And this, basically at ovulation, you end the follicular phase of this uterine cycle and you go into the luteal phase. Now, a couple of other minor details. Hey, how do we keep the hormonal system going for a bit here? The ovum, the egg cell, will eventually get picked up by the fimbriae, drawn into the fallopian tube. We'll get more into that the next chapter. But what happens, though, is what about the remaining cells that were providing the development, the maturation, the nurturing, the nest, sort of? Well, these remaining cells basically somewhat collapse and begin to kind of form, under the stimulation of luteinizing hormone, a structure that we call the corpus luteum. And by the way, what that means is yellow body. It kind of appears like a yellowish structure on the surface of the ovary. But what it does now is that its genes have been turned off and on, so that instead of making estrogens, it's making progesterone. And now, what's the role of progesterone? Well, progesterone is going to prepare the uterine lining. It's going to cause it to start building up. And it's going to enhance the uterine glands in that lining. And all of this is preparation for implantation and the sustaining of an implanted embryo. We call that implantation nidation. Again, I'm jumping a little bit ahead to the development chapter, but don't panic. I just want to get you somewhat familiarized. Now, what happens if we don't have fertilization? If you've already ovulated, and by the time you get to about 12 days, you're going to start noticing progesterone, estrogen levels fall. The corpus luteum is going to begin to disintegrate. This is referred to as involution. The tissue is going to be sort of taken over by fibroblasts. They're going to basically make this kind of pale scar tissue, and it's referred to as the Corpus Albicans. So if you start here, we go through the primordial ovarian follicle. You can see it right there. There's some follicular cells around it. Then we get into the primary ovarian follicle, and you can see the granulosa cells surrounding it. This is still the big egg. And then we get, with the development of thecal cells that specialize and support it, you have the formation of this thin membrane lining of the zona pellucida. You still have the granulosa cells that are going to help nurture it. And then you eventually get to the, excuse me, tertiary ovarian follicle stage. Now look, Here's the antrum. This is filled with follicular fluid. Here are some of the cells that are going to still stick to the oocyte, and they're going to be making up the corona radiata, okay? Then you have ovulation. You basically have this rupturing of the follicle and the expelling of the oocyte. And the cells that remain on the ovary are going to be part of this corpus luteum. The egg, the oocyte, with its corona radiata, they're going to be picked up by the fimbriae and go down into the uterine tubing. If you do not get fertilization, then what's going to happen is this tissue is going to basically begin to involute, and it's going to be replaced by basically scar tissue, okay? So we got all the primordial to primary to secondary to tertiary to ovulation, with the remaining tissue becoming the corpus luteum. If there is no fertilization, then it's the corpus albicans. Now, let's talk about the uterine tubes for a few minutes, okay? They're connected to the uterus. Now, the uterus is really kind of a hollow organ with thick muscular walls. The uterine tubes, and they're also known by several other names, oviducts or fallopian tubes, are connected to the uterine wall, and they extend laterally toward the ovary, all right? The uterine tube regions, you got to go through the names of them, okay? First off, the furthest one out, if you want to say distal, from the uterus, is the infundibulum. That's the funnel-shaped portion of the tube. The free end has these finger-like projections called fimbriae, and they help draw the ovulated egg into the uterine tube. The middle region is called the ampulla. It's kind of the longest segment. Usually this is where fertilization is going to occur. The sperm will meet the oocyte coming down through the tube. The isthmus is kind of a narrow medial segment that basically attaches to the walls of the uterus. Now, when we get to the uterus, it's kind of shaped like an inverted pear. In its position is referred to as anteflexon. It's located superior to the urinary bladder. Now, the segments of the uterus, you have the fundus, the body, the cervix. The fundus is that superior dome-shaped portion of the uterus. The body is the main portion of the uterus. Implantation of the embryo occurs along the wall of this region. The cervix, this is located at the inferior end of the uterus and projects into the superior end of the vagina. There are several different layers of the uterine wall that you need to be aware of. The perimetrium is kind of an incomplete serosa. It's continuous with the peritoneal surface, covers most of the uterine surface. This is on sort of the outer layer or outermost part of the uterus. The myometrium, this is a thick, smooth muscle layer. Sorry about the typo there. Provides much of the force to expel a fetus through the uterus and the vagina during delivery. And the innermost tissue lining is the endometrium. Now, this changes during the course of a monthly uterine cycle. Now, you have to keep in mind when we talk about uterine cycle, you're going to have that endometrial layer that's going to basically build up or recede down, break down. We'll get more into that in a few minutes. The uterine cavity structures. Well, there's a large superior chamber called the uterine cavity, and it's continuous with the isthmus of the uterine tube. We have the internal os, that's the opening that connects the uterine cavity to the cervical canal. The cervical canal is the passageway that connects the uterus to the vagina. And then we have the external os, which is this curved vaginal opening into the uterus. I want you to note also, when you look at the diagrams, you're going to see either attached through the broad ligament, you're going to see a lot of blood vessels, arteries and veins to the uterus. This is very, very important in supplying blood for nourishment of a fetus during pregnancy. As you can see here, here's the blood vessels I'm talking about. Here, of course, are the perimetrium, the thick layer of smooth muscle, the myometrium, and then we have that layer there, the endometrium, that changes over the course of a uterine cycle. The outside structures, you have the fundus, you have, of course, the body, and then you have, of course, the cervix there. This is where the external os is. This is the cervical canal. This would be considered the uterine cavity, and this, of course, is the internal os. All right. Now, the uterine cycle, otherwise referred to as the menstrual cycle, involves changes in the functional layer of the endometrium. The two layers of the endometrium we're focusing on are the basal layer and the functional layer. The basal layer is a layer maintained, and it restores the uterine epithelium during the proliferative phase. So it's almost as if to say, no matter what changes occur, basal layer is still there. It's the functional layer that contains a large tubular uterine glands. It's going to have an array of arteries also going through it. This layer is sloughed off during menstruation. When we talk about the branching of the uterine arteries also, we have the arcuate arteries, which encircle the endometrium, the radial arteries, which supply the endometrium, and then we have straight arteries and spiral arteries, which are extremely important. The straight arteries supply blood to the basal layer. It is the spiral arteries that supply blood to the functional layer. And during the menstruation, usually what happens is when you start having the crash in the estrogen and the progesterone layers, particularly progesterone, the spiral arteries will begin to spasm, and they will eventually choke off blood supply. And they're going to basically pinch off, and you're going to lose the tissue, but you will also lose some blood. Okay. And you can see here, here's the myometrium here, and you have the arcuate arteries through here. You have radial arteries which pass from the myometrium into the endometrium. You have these spiral arteries here which go up, and you notice that the uterine glands are here. A better way to look at it is here. Here is a spiral artery going up into the functional layer. Straight arteries, as you can see, they don't curl, they're not coiled. This is part of the area you would call the basal layer. Okay? When you have the fall in the amount of progesterone, these layers will slowly disintegrate because these blood vessels will choke off and spasm and close up. You'll lose some blood, but you won't lose an extensive amount, usually. And what will happen is all of this tissue will be lost. But let's talk about the full picture of a uterine cycle. It's also referred to as the menstrual cycle. Basically, what we're talking about is this refers to the cyclical changes that occur in the endometrium over the entire menstrual cycle. When I'm talking about the menstrual cycle, by the way, everybody thinks, oh, 28 days. Yeah, that's average. There's some women, and I'm sure there might be a few of my students in there kind of nodding right now. Their range is anywhere from 21 to 35 days. If they sat throughout the whole year, they could write this down. Now, there's some women that have, how can I say this? They have cyclical dysrhythmias, and basically they may say, well, I had my cycle and it was fairly normal, and I had another one that I didn't have until 3 months later. And there may be a whole host of reasons that basically, a doctor could perhaps explain. So let's just take a typical 28-day cycle, okay? First, you have the menstrual phase. That's approximately 1 to 5 days. You have the generation of the endometrial glands and the blood vessels. Basically, this is where the functional zone is breaking down. The endometrial tissue and the blood pass out of the vagina as menstrual flow, the menses. The proliferative phase, which is approximately 6 to 14 days. This is where the basilar zone gives rise to a new functional zone. Estrogens produced by the ovarian follicle induce endometrial thickening and enhance blood vessels in this zone. Now, the secretory phase, this is where you get from about 15 to 28 days. This is the final stage of the uterine cycle. It occurs to prepare the endometrium for implantation. The uterine glands are large, they're complex, they secrete glycoproteins and other nutrients. The arteries will supply the early embryo with maternal blood or resources from the maternal blood, such as oxygen and nutrients, etc. The activities are supported by progesterone produced by the corpus luteum. Okay? The menstrual cycle will begin at puberty. The term menarche is really a young woman's first cycle. It typically will occur somewhere in the age of 11 to 12. The term menopause refers to the end of menstruation. This is an age range somewhere between 45 to 55. Some women have had it earlier. Some women have had it a little bit later. There are situations that cause interruptions of menstrual cycles: illness, stress, starvation, medications, contraceptives, and of course, pregnancy. There has been those situations where individuals have said, "Oh, well, we had the baby. The woman is not still starting her menstrual cycle, so they can have intercourse again. And surprise, she was fertile." And this is where you get some of the situations where they call them the Irish twins. In other words, babies born only 13 months apart. Okay. Or you could say maybe 12 months apart. Okay. But what I'm just saying to you is that there are situations where individuals will become what we call amenorrheic, no menstrual cycles, and it's not of their own doing. Okay. And you can see here's the menstrual phases. You can see where the uterine glands have been breaking down. The basal layer is still here. This is the uterine cavity. If we go from here to the proliferative phase, you have the basal layer. Notice it's gone down somewhat because the functional layer has grown upwards, and you have now more development of the functional layers, uterine glands. You would have more blood supply going there, etc. When we get to the secretory phase, you will know an extensive network of uterine glands, and the layer is much, much thicker, such that the basal layer would be much lower below here. And this would make the conditions proper for nidation, the implantation of a fertilized embryo, or excuse me, of an embryo, no longer calling it a fertilized egg. That only goes for a zygote. It's only the one cell stage. By the time you're getting in there, you're many cells as an embryo. Now, the vagina opens into the vestibule. The vagina is an elastic muscular tube extending from the cervix to the vestibule. The vaginal canal is an internal passageway. The vaginal fornix is a shallow recess in the vagina surrounding the tip of the cervix. And the rugae, this is the vaginal lining folds. These vaginal lining are composed of non-keratinized, stratified, squamous epithelium. Okay, and you can see all of this right here. And partly this is non-keratinized to handle, and it's stratified also, to handle the rigors of sexual intercourse, but at the same time to allow for flexibility of both the muscular and the tubular structure. Now we go from here to the external genitalia. And this includes the mons pubis, which is a mound of fat and skin that covers the symphysis pubis. After puberty, the skin is covered with pubic hair. The labia majora is two fatty skin folds covered by a pubic hair. They project posteriorly from the mons pubis. The labia minora are two smaller skin folds. They're located within the borders of the labia majora. They're highly vascularized and they are hairless. Then you have, of course, the vestibule, which is the space enclosed by the labia minora, and the clitoris, which is the cylindrical structure composed of erectile tissue located anterior to the vestibule. Vestibular glands. There's two major types. The greater vestibular glands, otherwise known as Bartholin's glands, discharge some fluid during sexual arousal, and they discharge it usually into the vestibule. The lesser vestibular glands discharge secretions onto the exposed vestibular surfaces, basically keep it moist, as you can see here. So here would be the bulb of the vestibule, the vaginal entrance over here, the greater vestibular glands lower here, the labia majora and the labia minora up here. So this is the majors part. This is the mons pubis, okay? And the clitoris structure is right over here. Okay. Now we move over to the breast, the mammary gland, because that is critical to secrete milk. Interestingly enough, mammary glands are modified sweat glands. Their function is to produce milk that provides nourishment for a baby after birth. Now, when we get into next class, I do encourage you to pick up the handout on the value of the initial milk that comes from the mammary glands, as a lot of it enhances the immunity for the newborn. The term lactation, this is milk production, and it's controlled by the hormones prolactin and oxytocin. Prolactin usually stimulates the production of the milk. Oxytocin will play a role in the triggering of the milk release, the milk letdown reflex. But again, that's something that we'll get further into when we get into the next chapter. The structures that make up the breast include the suspensory ligaments, which support the gland and travel from the skin to the deep fascia. We have lobes in there, and each lobe is made up of lobules. The lobes in the range of 15 to 20, they're divided by fat and connective tissues. What lobules are really are the clusters of glandular cells that produce milk. Now, the lactiferous duct, this is a structure that milk is secreted into these ducts and eventually drains into the lactiferous duct, which will converge at the nipple. The areolar is the external structure. It's darkly pigmented. It's circular area of the skin. And usually in the center of the areolar is the nipple, which is the structure projecting from that center area. And it's the site to suckling by the infant, which will trigger the reflex to release milk. We have, before we get to the release of milk, we have these lactiferous sinuses. These are storage sites of milk prior to breastfeeding. And the duct expands to form a sinus structure, as you can see here. So here we have the lobules, okay? This makes up an entire lobe structure here. You have the areola, the nipple. You have the lactiferous sinus, the lactiferous duct, the secretory alveoli. You have the suspensory ligaments, and of course, the accompanying adipose tissue. The next area we're going to be discussing is the ovarian and uterine cycles and how they're regulated by the hormones of the hypothalamus, the pituitary gland, and the ovaries. I strongly encourage you to take time to review the diagrams, which I will put up on the slides in a minute, but they're also available on pages 1024, 1025 of your text. Let's start off first and foremost with the hypothalamus, okay? It's going to produce and release gonadotropin releasing hormone, okay? So GnRH. Now, this is going to act as a trigger to release in the pituitary the following two hormones: follicle stimulating hormone, FSH, luteinizing hormone, LH. Now, FSH acts on the ovarian follicle development as well as on the secretion of inhibin and the secretion of estrogens. Keep in mind that inhibin has a negative feedback effect on the secretion of FSH. Now, LH acts on the meiosis 1 completion, on ovulation, and on the formation of the corpus luteum. Now, when we get to the ovary and the corpus luteum, this is where we get into some other hormones produced. The ovary makes estrogens. Estradiol happens to be the dominant type of estrogen that's produced just prior to ovulation. And you can see all of this here, but let me jump ahead and come back. The corpus luteum, though, turns around and starts making progesterone. Now, if you can see here, GnRH, the release of it, will act on the secretion of FSH, which plays a role in ovarian follicle development, secretion of inhibin, and secretion of estrogens. Inhibin will play a negative feedback back on the release of FSH. Now, interestingly enough, before day 10, there's a suppression of the secretion of luteinizing hormone here. After day 10, there's a stimulation of it. This is all involved in part with the corpus luteum after ovulation, but also beforehand, estrogens play a role in an inhibitory way to the LH. Now, note down here also that what you have are a variety of physiological effects on the CNS, bone muscle growth development, establishment and maintenance of female secondary sex characteristics, maintenance of accessory glands and organs, and the stimulation of endometrial growth and secretion. This is all being influenced by estrogens. But let's not forget the other side of the coin, and that is that we have the release of luteinizing hormone, which is also under the influence of GnRH, which its secretion is involved with meiosis 1 completion, ovulation, and corpus luteum formation. Corpus luteum formation leads to the secretion of progesterones. It also will play a role in the stimulation of endometrial growth and secretion. All of this side is really the luteal phase of the ovarian cycle. All of this side is on the follicular phase. Okay, one other question. Where do we have negative feedback effects? The secretion of progesterone feeds right back all the way to the top. It suppresses GnRH. Now, let me help you understand this just a little bit again going into the next chapter. When you start producing progesterones, you don't want to be ovulating again, because if you have sustained secretion of progesterones, that's going to play a key role in maintaining the development of the implanted embryo in the uterine lining. In essence, you've got a developing embryo inside. You don't need any more releases or ovulations at the time. Okay? That's why progesterone has such an impact. The impact of inhibin is to modulate also the secretion of FSH so that it turns it up and turns it down depending on the state of ovulation at the time. Okay? Now, I've talked about inhibin being negative feedback. Okay. I've talked about progesterone being negative feedback to GnRH. I want you to note, and in a minute I'm going to show you the chart, the spikes that you see with LH, FSH, and GnRH at about the time of ovulation. The interesting thing is that progesterone has a spike later during the secretory phase. Now, there are two other little concepts I wanted to bring up. One of them is basal body temperature. This is the resting body temperature measured at awakening in the morning. Roughly, it's going to be 0.5 degrees Fahrenheit lower during the luteal phase due to progesterone. This brings us back later on to a very brief discussion on the rhythm method. Some people will state it works, some people will state no, some will talk about the error rate. And when I say instead of the failure rate, it's really an error in measuring the basal body temperature plus several other factors. But we'll move into that in a minute. You have to keep in mind, as I said, that estrogens play a role in secondary sex characteristics as well as physiological systems. Here we notice FSH, and it has a quick spike just before ovulation. So does GnRH. And by the way, these are all not just continuous levels. The unusual behavior that you see with hormones from the hypothalamic and the pituitary is they're in pulses. So many pulses per day, as opposed to just one continuous flow. But what you're going to see just before the time of ovulation is a spike in LH, a spike in FSH, and GnRH. At the same time, you're going to have this massive spike in estrogens, and then ovulation will start going down. Inhibin has this spike just afterwards, which is going to cool off, and that accounts for the trough you see in FSH. Progesterone, interestingly enough, really rises, but it rises after ovulation and stays there for a sustained period of time. If fertilization occurs, if implantation occurs, this would not drop down. It would maintain itself at a certain level because it would sustain the secretory lining, the functional lining, okay? Notice the basal body temperature just before you hit ovulation drops about 0.5 degree. And then you have this other 0.5 degree difference, correction here. You have a slight decrease here just at about the time of ovulation, and then just after ovulation you have this pickup, and the temperature difference is about 0.5 degrees Fahrenheit, okay? And it stays there for quite some time unless there is no fertilization. A lot of couples who had problems conceiving used the method to determine when fertility occurred, then of course had sexual intercourse, and then of course had a successful and healthy pregnancy. Now, let's also talk about pregnancy from the perspective of birth control strategies. They vary in effectiveness and associated risks. Male condom. I should put this in the male reproductive system, but I'm going to put it in here now. It's basically a barrier. It's a latex protection over the penis to prevent the transfer of sperm and the transfer of sexually transmitted diseases. Basically, if done properly, there's actually one that's even more interesting, and it is called the cervical cap, which literally is a smaller, somewhat condom, but instead it goes over the cervix of the woman, and it's removed later on. It's similar in its approach to the diaphragm, but I'll get into this in a bit. The birth control pills, otherwise known as oral contraceptives, they prevent either ovulation or implantation of the embryo. The risks include blood clots, heart attacks, stroke in women over 35. A lot of doctors, if they know that they have a patient that is getting over 35, maybe overweight, and one of the other factors they'll sometimes sit there and say is if they're smoking, they'll be sitting there saying, I want to change you off oral contraceptives, something else. And of course, then they get into a big to-do about this. I've known more than one friend that had this argument with their doctor. The next one is the intrauterine device, otherwise known as IUD. You have the copper T, or you have some other ones, and they're hormonal in their release. These usually prevent the sperm entry and its transport from the uterus into the fallopian tubes, or the hormonal release will prevent ovulation. Although there is some thought that what the copper T also will do is that will irritate the lining to prevent nidation of the fertilized, of the developing embryo into the uterine wall. The diaphragm, it's a shallow silicone dome. It's fitted over the cervix. Usually what they have is spermicide as a gel applied on the inside, and it's basically another type of barrier to prevent sperm entry into the uterus. Now, progesterone-only birth control pills. What you usually have is either a injection or a low-dose progesterone pill. Now, here's the principle. If it's progesterone, it's basically fooling the body to think that it's pregnant when it's not, and therefore it suppresses ovulation, and so this basically works for a while. Now, there are a couple of really serious caveats here. A lot of women are told by their doctor, if you're on this pill, the low dose progesterone, you have to understand that you will gain weight. You may also have some acne. You may also, if you skip one pill, could get pregnant, because it's really almost like, thinking about it this way, like a bird flying very close to the waters, to the basically to the waves of the water. If you lose one pill, the level falls below it, and then ovulation can start again. The other problems are that it may cause a delayed return to fertility. So if the couple decides they want to get off of this and have a child, it may be up to 18 months before the body is restored to its normal function. The rhythm method is a method that combines basal body temperature and other indications of fertility. One of them is cervical mucus thickness. During the fertile period, the cervical mucus changes viscosity, and that may also be, plus the basal body temperature, a sign. Some people call that the same thing as like sitting there and counting the calendar, and that's not really so. Counting the calendar is under the guise that you think, every month is going to be 28 days. But as I've said to you before, people have variations there. The value to the rhythm method also is that for couples that have been trying to get fertile, it will give them the indication of when they are actually fertile, and then they can decide if they want to abstain or have intercourse and have the child. A postcoital contraception, otherwise referred to as Plan B, this is a morning after contraceptive, and it prevents the fertilized embryo from implantation. It's usually hormonal in its approach. Then we have surgical sterilization. Now, I mentioned vasectomy for the males last lecture, I believe. Tubal ligation is the process in where they basically will cut, they may tie off, they may ablate, which basically means cauterizing the ends of the cut tubes. This prevents sperm passage or fertilization. Usually the process is very non-invasive as much. There's still a little bit of invasiveness, but I mean, they do it laparoscopically, so it's done very quickly, and the person can go home the next day. And these are some of the models that you see here. And usually for the uterine tubes, they can see this with a laparoscope. They will then clip a small piece of the tubing out. And one has to be aware of this. If they don't cauterize and don't properly tie, there have been cases where some young women have been pregnant after this procedure. Sometimes, though, it can lead to what's called an ectopic pregnancy. An ectopic means out of the normal place. An ectopic pregnancy might be outside in the body cavity out here, where it implants and begins to start forming development. Ectopic might be a high risk one that's called a cervical pregnancy, where the placenta forms right here. Or ectopic may be where it forms in the tube. If it forms in the tube, the usual strategy is you have to sacrifice the pregnancy and you have to remove the tube, because otherwise what it'll do is rupture the tube, and in the olden days before surgery, etc., it would have killed the mother and would have killed the developing fetus as well. Okay, reproductive system disorders. They're relatively common, often deadly. We have to be mindful of some of them. We talk about breast cysts, and these are inflamed breast lobules that become walled off by scar tissue. This can form clusters of these cysts, and this is where we get fibrocystic disease. So it can be felt in the breast as a fibrous mass. It's still, though, a benign condition. But breast cancer, usually that's malignant metastasizing tumor. The risk factors include a family history of breast cancer, the first pregnancy after the age of 30, the early menarche or late menopause. And there have been genes that have been isolated that seem to cause a high risk of the breast cancer. They've been called the breast cancer genes, but to give you a perspective, this is not one particular site on a particular, like the BRCA1. There are believed to be 300 hot spots, in other words, sites on that entire gene where a mutation can lead to a cancer. So that's what makes it a little bit tougher to identify accurately everything if you have sort of a gene. You just have to perhaps be much more monitored as opposed to believing that you will eventually get cancer. Ovarian cancer, these are cancers caused by ovarian tissue or abnormal oocytes. If we talk about carcinomas, these are cancers derived from epithelial cells. Cervical cancer, these are cancers of the cervix. Now, one of the things that happens with the cervix is that's where we have the Pap test from papilloma. And what the doctor did was he was the first one to take cell scrapings and notice if there were any abnormalities. If they were, that suggested a risk of, from the cervical scrapings, of the possibility of cancer. And they looked at the different developmental stages of what these cells were. Interestingly enough, a virus called the human papillomavirus, HPV, is a virus that's found to be not only a sexually transmitted disease, but it can cause 75% of the cervical cancers. And there are vaccines out there to basically immunize an individual against 4 of the substrains of the HPV virus. The problem is that you could end up with antigenic shift down the road, or I should say target shift, where other versions could then become prominent depending. When we talk about sexually transmitted infections, it was formerly called STDs. There are a variety of diseases that are out there. You may have the opportunity to take this in medical microbiology, but just to give you a few of them, the bacterial ones, syphilis, gonorrhea, chlamydia. Some of them are as not easy as to treat now as they were. There's a lot of drug resistance in certain strains of gonorrhea now that has gotten a lot of concern worldwide. Viral STDs include, of course, HIV, hepatitis B, and hepatitis C. The protozoal ones are trichomoniasis, and this is the trick. It's an actual protozoal parasite. And then we have the fungal types. One of them is candidiasis, and certain candida that will get in and proliferate, particularly in the vaginal area. Some of these, unfortunately, will render an individual, a woman, sterile. Chlamydia, for example, can go into the tubes of the fallopian tubes, cause great proliferation, and then basically close up the tubes, filling them with scar tissue. So that's why some of these diseases have to be dealt with very seriously, because they can lead to unforeseen effects down the road, affecting the person's fertility. Here is a situation we have a breast cancer with calcification. So in other words, as the cells have formed, they begin to form calcifications here. Oh, wait a minute. Okay, here we go. Sorry about that. So we have ovarian carcinoma. This is the size of a normal ovary, and what has happened is there is a cancer inside of this ovary. It may be caused by some of the epithelial cells, or it may be caused by abnormal oocyte cells. But that usually would lead to the removal of the ovary, and depending on how and if it has metastasized, to perhaps take both reproductive organs out, both ovaries out. This is an example of cancerous tissue on the vaginal wall, as you can see here. Here is the external os here, and you can see that this is somewhat spreading throughout here and would have to be basically removed. Okay, so we have completed this chapter. I would encourage you to review the chapter, to go over the chapter review on pages 1031, 1033. Complete the review questions 1034, 1035, and review the sheets on pages 1030. We're going to go in next week with chapter 27, Development and Inheritance, and I hope you all have a nice day.